So birth of Mendel took place on July 22nd. Okay. It was in the year 1822. He was born in a village called Sicilian village. The village name was Sicilian village. Okay. And he moved to a monastery. It's a place. Okay. He moved to a monastery. And after that, for his studies, he went to the University of Vienna. Okay. That is there in class 10 book. Mendel University of Vienna. He studied there, and then again he returned to monastery. Okay. He returned to monastery and he joined as a teacher in monastery. Okay. So here he didn't have any idea about maths or anything. He studied and then he experimented. Okay. He died in 1884. This is not asked, but just consider that as important because most of the times years have been asked from this chapter in this topic, in this particular topic. Okay. So work on Pisum sativa. Pisum sativa is a scientific name. And always remember, whenever you write Pisum sativa or the scientific names, you must underline it. Okay. So Pisum sativa. Separately you have to underline, and if it continues, the starting letter of the second word must be small. Yeah, it is very necessary. Only when you understand it, there is a very big chapter based on it in first year. How you must write scientific names and all. Okay, rules of scientific names and stuff. It is also known as garden bee. Okay, and it is an edible bee. We can consume it, right? So it is an edible bee. So this experiment, whatever he had conducted on Pisum sativa, it is having a tea plant. He conducted it in the year 1856. He conducted it between these. Okay, 1856 to 1863. Between those years. Okay, so the experiment was conducted between 1856 to 1863. Okay, and it took 7 years to conduct these experiments. Okay, the gap between 1856 to 63 was 7 years. Fine. And this experiment, whatever he conducted, that is known as hybridization experiments. Okay, whichever experiments he conducted on Pisum sativa, those experiments are known as hybridization experiments. Okay. So reasons for Mendel's success. Why did Mendel get succeeded in this thing? And why was he called as father of genetics? Okay. So then first one was he applied mathematics in biology. Okay. And he got ratios and all right. So that is why he applied maths, and he applied statistics also. Okay. Statistics. Statistics and probability is a part of maths. Statistics is also a part of maths. Okay. So he applied mathematics, statistical analysis also. Okay. And then he kept records of all the experiments which he did. Like he do the experiment and then he didn't just leave it like that. He wrote all his observations. He wrote all the results. Now it stored it. Okay. And then the sample size was huge, so the credibility of result was more. Suppose if he was asked to collect the samples with height in the station, okay, like everybody's height or the average height, average tallest height or average height, which were like, okay. So I would take yours, only 4 of us. Okay. So among us, taller one will be sir. Okay, because we are only 4 samples. Okay, but if I consider the entire tuition of kids, I'll get even more people, right, with the same height or with other different heights. Yeah. Okay, so that is what he considered large sample. Sample size is huge. Okay, so that he got more, credible results. Not massive results, precise results. He got more precise results. Okay, and then he experimented it, and most of the scientists, what they do is they either experiment on few things or they don't experiment and then just by words they tell that they are using it. But he conducted experiments and he also gave theory of it, he explained it. Okay, that is theoretical inference. Understood? Cool. And then successive generations, like he took more and more generations. Okay, so that you got same results. Like suppose see if you take wrinkle and round. Okay, then you cross between them. Okay, so he got the same result. For that, the first cross means suppose round wrinkle. Okay, so he took round dominant and recessive and he crossed between this. So he got F1 generation as R and R. Okay, and when he did selecting between this, he'll get 1:2:1 ratio. Okay, and even if we take tall plant, small plant, even if we cross between this, the ratio will be same. Right? So that is what it means. Successive generations, you got same ratios, same results. Okay, so that non-substantial, it made sense. Okay, all these made sense, right? Because he experimented it and he explained how it's happened. Right? So that is what it means. And he published his work in 1865. This is a very important question. Okay. This is PYQ. That's why I put it on the name. It's like repeated most of 3 times or 4 times. I don't know how the length will be when you write the exam, but this is the most popular. Okay. So Mendel published his work in 1865 and the experimental years were 1856 to 1863. Okay. And Mendel's work remained unrecognized. See what happened is Mendel experimented and he published his work in 1865. Okay. And even after that, he didn't get recognized. He passed away in 1863. Oh sorry. Yeah. He passed away in 1884. Okay. So until he passed away, until he died, he didn't get recognition for his work. Okay. Like because of few factors, there were no communication. His work remained unrecognized. That is nothing but because whenever he was alive, he wasn't able to see his success. Okay. So the first factor was communication was not easy. Like right now we didn't have social media at that time, right? So communication was very difficult. Okay, and no internet connections. Internet also plays a role in this. Okay, internet. And then he mentioned something called low factor, which is nothing but GC. Gene was identified in the year 1909. Okay, but he suggested his work in 1865. Right? So at this time there was no term called gene and no one was introduced to a term called gene. Okay, but he told that something is passing from generation to generation. Okay, and that something he named it as fact. Okay. And then applying mathematics. Take it out. Mathematics. Biology didn't make sense. And no clue. I asked you to leave 1 point for 2 lines.
Yeah, 1.2 lines.
What happened? Did you fit in that?
It will fit. What's this to be?
I'll give you something to fit in that. Okay. Right. Okay, what? Factor. No. Physical. No physical proof for factor. his experiments to be unrecognized. Okay, those factors are communication was not very easy in those times,
Yeah.
right? And internet was not there. Huh? Okay, internet was not there. That is one. And factor, which is referred now as gene. Okay, he told that something is passing from one generation to another generation. He didn't know that it was gene. Okay, he told that something he named it on himself as a factor. Okay, so that something was named as factor. And using of mathematics in bio, people would be like numericals in bio, who would do it math. So people didn't accept this. Okay, and no proof for this factor is passing from generation to generation. That's what he told, right? But there was no physical proof for it. Okay, so this thing was not accepted by his co-workers, and most of it was not accepted. These were the drawbacks of it. Okay, and Mendel's work was re-recognized in the year 1909. Wait, 1909. Did you write 19,000? Write in 19,000. 1900. Okay. So they conducted experiments. Okay. They conducted it independently. They didn't combine. All three scientists were not. They didn't combine and do an experiment on the same thing. They conducted it independently. Okay. So on themselves. Okay. So first scientist was Karl Correns. Okay. He is from Germany. Okay. And second scientist was Testor Mark. Okay. He is from Austria. And third scientist is Rutger Veres. He is from Holland. Okay. Holland. So these three, they published their work, whatever they concluded or the results, in a book called Flora. Okay. This is a book. In a book called Flora, they published it in the year 1901. After they published it, people got to know and people read this. Okay, people read this and then they concluded that even Mendel also had suggested this years ago. Right? So that is why they gave him the name as father of genetics. Fine? And why did Mendel choose Physium satire? That is because there are reasons in Western also. Few. Okay. That is because it has very short lifespan. What does it do when it has short lifespan? What impact does it play? He can observe more generations within a short lifespan, within a short period of time. Okay. So it had 3 to 4 months of lifespan, and it was easy to grow in labs. It was easy to grow in labs. It was easy to grow everywhere. Okay. So you could grow it easily in labs also. That was one thing. And large number of offsprings or seeds were produced. Okay. So one plant used to produce like 4 offsprings, right? One selfing and all. So that is why it used to produce large number of offsprings or seeds. Okay. And it was bisexual. Bisexual is nothing but cross pollination also. Stigma and pistil and this thing, stamen will be present in the same flower. That is what it means by bisexual, right? So there is something in NCERT called as emasculation. Emasculation is when, see, suppose if this is a flower. Okay, this is the flower. This is You want to get an hybrid fruit rather than getting a papaya from a papaya tree. You want to cross between papaya. Take as an example, okay? Papaya and watermelon. Say watermelon, okay. So if the flower is bisexual, this pollen grains which will have grown, when it matures, it will fall on this stigma, right? So it will fall on the stigma, and the pollination will happen, and growth will happen, then it will form seed, then it will form fruit, right? So if you don't understand anything, tell. Okay? You have any doubts? No. Okay. So this thing happens, right? But this happens in case of papaya. Papaya plants ka flower only basis. Okay. So this papaya pollen grains only will fall on the papaya stem. Right. But I want to cross between watermelon and papaya.
So watermelon
Pollen grains.
pollen grains.
So what I do is before this thing male part grow, I will take it off. ಅದು ಮೆಚೂರ್ ಆಗೋಕ್ಕೆ ಮುಂಚೆನೇ ನಾನು ಕಟ್ ಮಾಡಿ ತೆಗೆದುಬಿಡ್ತೀನಿ ಆಯಿತಾ. I'll take it out and then on this ovary, due to wind and all other pollen grains might come and land, right? So that is why I'll cover it with a bag. Okay. And then whenever I find a pollen grain of watermelon, okay, I'll take this bag, I'll put the watermelon pollen grain in the stigma. Stigma.
This is stigma.
Stigma.
Stigma itself.
That's what I do with stigma right now.
Okay.
This is stigma.
Yeah, stigma.
Okay. So I will put pollen grains on this. That is emasculation. Okay. See, it's given removal of anthers, emasculation. Okay, that process will happen. Is everything going out of a brain? Okay. So bisexual flower, right? So I can cross pollinate also using emasculation, and I can do it self pollination also through that. Right? So that was an advantage. Okay. And self pollination, there is something called as true breeding and pure line. Pure line. Okay. This is nothing but when I self pollinate TT and TT. Okay. When I self pollinate, I'll get offspring with TT and TT. Okay. So the first parent which I took was big T, small t.
Why?
What do you mean by? Mendel did that on you know. Okay. Dominant and recessive. Okay. So when he cross pollinated this two, he got this one. T and T dominate. Cross pollination ಮಾಡಾಗ, okay, crossing ಮಾಡಾಗ. So ಇದನ್ನ ನಾನು self pollinate ಮಾಡ್ತೀನಿ. ನಾನಲ್ಲ ಮೆಂಡಲ್ ಮಾಡ್ತಾನೆ. ಆಯ್ತಾ? So this TT self pollinate ಮಾಡಾಗ ಇಲ್ಲಿ TT capital T small t. Right? Another capital T small t also will be there. I'll explain you that. Don't worry. Okay? I'm just giving you how it works out. Okay? So see, this was my first parents which I took. Right? So this TT is resembling this now only, probably. Right? And this TT is resembling this only. This TT is resembling this. And this one also is resembling this. That is nothing but 3 are tall and 1 is dwarf. Okay? 3 are tall and 1 is dwarf. So it resembles parents only, no? Middle ka size is not coming. Each ka size and all is not coming, right? So that is what it says. Produces offspring with similar traits of parents. Parent was tall and parent was short. So here the offspring will be short, tall, short. Only these two can be there. No medium thing around here. Okay, that is what is true breeding. It happens only in self-pollination. Okay, and contrasting traits. That is nothing.
If the parent is short, if the parent is short, the seed will be short.
ಎರಡು short tomato, short. Okay. So this contrasting trait is nothing. It says the same thing. Tall or short. Okay. There is no middle size. That is what it made a pyroxylene the advantage. Okay. You understood this?
Yes.
Do you have any doubts?
Emasculation means cross-breeding of.
Emasculation is not cross-breeding. Just remember that emasculation is removing of anther from the flower and selecting the pollen grains whichever I want to pollinate to the stigma, and then getting removing the anther before it matures. Okay, and then covering the stigma with a bag of plastic or anything, okay, or paper. Okay, and then whenever I get the gold grains, whichever I want to put on stigma, I remove the bag, I put it. That's all. Pollination will happen. Why I put the bag? Because I didn't want any other pollen grains to come. Okay, by removing. That's why I put the bag. Understand? What is the importance of a short life period for the organism, very fast, for the organism chosen as an
Organism.
chosen chosen
Okay.
as an experimental material
Mental material.
experimental material by a geneticist 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 for conduction of studies on pattern of inheritance. What is the answer? On pattern of inheritance.
What is the 3 to 4 hours.
What is the importance of short life period? What is the importance of that?
It must be nothing. It needs to be the generation, the ratio must be same.
I just told you the reason.
The same thing. The action thing. Those things must be same. They must be
Explain what you say.
the ratio
Mm-hmm.
of the pattern of the cross.
Why is it 2 to 3 months? Why is the plant grows only for 2 to 3 months, right? What is the importance of that? Like, what does it gain? What is the difference? Like, when it is 2 to 3 months, when the lifespan is 2 to 3 months, why is it 2 to 3 months? Why did he choose it?
Because the generations of those lifespan is same, and the ratio, the thing is same as when they crossbreed, and
It's just a very simple answer.
you can see more generations.
That's all. Yeah. That's an unacademic test question. Gregor John Mendel was an Austrian monk, right? Austrian monk.
That's what
Point, important point. Gregor John Mendel was an Austrian monk. Take definition of homozygous. The organism homozygous, we have two, okay, we have two types. That is nothing but double heterozygous and double homozygous. See, if TT, okay, this is same and this is same, right? So this is double homozygous. Okay, and suppose it is TT and TT. Take a TT and RT. Okay, this one is heterozygous and this one is also heterozygous. Take this one also as round. Tall, round. Okay, round wrinkle because it is R, small r, wrinkle, tall wrinkle. Okay, so this is double homologous because T and T is same, R and R is same, right? And this is double heterozygous. Take it off fast. Have to complete so much. Okay. So take it down. Rhizome sativum. It is a diploid flower or diploid plant. It's a diploid plant. Okay. And then it is sexual reproduction is possible. So sexually and it has two ends. In humans we have 246 chromosomes, right?
Yes.
But in Rhizome sativum it's only 14. Yeah, diploid is 14. So haploid will be 7. Okay, na? So diploid is 14. Take one. this order, okay? Remember it in this order only. In this order only. No exchanging. Okay, I'll tell you why. Seven, 1, 1, 4, 5, 4, 4. This is 4. This is 4. This is 5. This is 4. This is 1. This is 1. This is 10. Height. Okay. So here we have dominant and recessive. And recessive. So seed shape, dominant is dominant, and we represent it by capital R. Take it, blink it, and we represent it by R. Seed color, dominant is yellow, and we represent it by Y. Recessive is green, and we represent it by small y. Flower color, violet, green, and white. Small, this is capital UK. This is small. Heart shape, full. Constant. Font color green, yellow. Flower position axial, A, terminal, small. Tall T, dwarf, small t. Now, okay. So, take on this question. We took those numbers. Question: how many pairs of contrasting characters? Pairs of contrasting characters in pea plants were studied by Mendel in his experience. Options: 8, first option 8, second option 7, third option 5, fourth option 6. What's the answer? Not 7. Next question. In his classic composition, seed color, pod length. What's the answer? Okay. Done. Next question. Just tell me the answer to this. In organism with two I'll give you time to write. Just tell me the answer. Okay. Listen to the question. I'll give you options. Tell me the answer. In organism with two identical alleles is identical alleles. I'll give you options. Two identical alleles is dominant. Option 1 is dominant. Option 2 is hybrid. Option 3 is heterozygous. Option 4 is homozygous. Take it down. In organisms with two identical alleles is two identical alleles is option A dominant, option B hybrid, option C heterozygous, option 4 homozygous. So, here, these numbers, okay, in Pisum set diagram, I will write as P plant. In P plant, we have 7 characters, which is nothing but 7 pairs of chromosomes, right? I told you that 2n is 14, right? So that is 9 or 14 chromosomes are present. Right. So when I put it in the form of pairs, I'll get 7 pairs. So out of write it out. Out of 7 chromosomes, see, 1 is 7. Okay. These are the chromosome numbers. Okay. 7, 1, 4, 5. Okay. So these traits, whatever is there in the P plant, it's only present on 4 chromosomes. Only 4 chromosomes. Rest 3 are empty. It's not like it's empty, it will have other characters.